Hi everyone, how are you? 8 a.m., lecture number 5. So thank you for coming, thank you for keeping up. We're not too far from the midterm. Check the syllabus, we are close. Make sure you get the most from the course in discussion sections, in these flipped classroom sessions, in office hours. I'm having office hours today, but IAs have office hours as well. So make sure you ask all your questions and you are understanding deeply the concepts. Okay, so what we will do today, we will start with a clicker. And so I need a reminder from you. Did we do this question last lecture? I don't remember if we got to this one. Okay, so we'll start with the next one. And I just wanna make sure this is, I will pull it and okay, so you can start, I believe. And I guess that I need to hold on a second. Actually, why don't you start answering these questions again on your own, and we will go quickly afterwards, because now I realize I need to connect this. And Marina, I got an adapter. But yeah, do this one and the next one and the next one all on your own, and we will go quickly afterwards, okay? And we will have to start again because I need to connect the base so people that have the physical clicker can vote, okay? So these ones will not count, and I will start again. Okay. Thank you, Josh. So I believe everyone can vote now. So if you could please vote, so I can stop the poll, and we can go through this question together. So please vote. Is anyone having trouble with the clicker? Anyone not detecting the base or something? Okay, so everyone voted, correct? Okay, so I will stop it now, and this one will count, okay? And so the question goes as, which one is false about the effectors of adaptive immunity? So most of you answer C, and so we'll see if that's correct, but there were other answers as well. So antibodies can activate complement, can opsonize antigens, and or neutralize pathogens. This is all correct. Any question? Okay. So CD8 T cells are specialized for recognizing and killing virally infected cells. That's totally true. They'll also kill tumor cells, right? When the tumor has a mutation that change the amino acid sequence in the self-protein, now it becomes, this mutation becomes a foreign protein, makes the protein becomes foreign, and so it's recognized by CD8 T cells as well when presented in MHC class 1. Anyway, so this is true. B is true. C says the adaptive immune response empowers the effector mechanisms of the innate immune system, except for antibodies which never act via innate components. And most of you answer this one as false. Anyone wants to explain why? Yes, Marina. Exactly. It can act through the innate immune system using complements, right? Which other way the antibodies use the innate immune system or innate cells to destroy pathogens? Yes. Opsonization, right? For macrophages, macrophages will learn are part of the innate immune cells, so that's the other cell. So this part that says except for antibodies which never act via innate components is wrong. And we will learn later that there is another way that is through natural killer cells, okay? But we'll learn that later. Any question about that? So this is false for sure. So let's look at other ones that maybe were confusing. An antibody secreted by a particular plasma cells share the same antigen specificity than the BCR on the B cell precursor that generated such plasma cells. So some of you consider this one as false and some true. And so what do you think? Do you want to bring it up to the group? There is obvious some confusion, right? Why? Yeah.
When you have a cell with a PCR, it eventually secretes it, so that PCR that was secreted is the antibody. Right?
Exactly.
So it's exactly the same.
It's exactly the same. There's the same antigen specificity. It's not. If the question were exactly the same, it's not exactly the same because it doesn't have the transmembrane region. Right? It's now a soluble protein. But all the rest is the same, except for the transmembrane region. And so that means that the part of the antibody that binds the antigen that you learned already in lecture 5, pre-recorded, what is the part of the antibody that binds the antigen? Is in which part of the antibody, the antigen binding site? The CDRs, the hypervariable regions in the variable region of the antibodies, they are the CDRs that are hypervariable. And that binds the antigen. And which part of the antigen will bind? The epitope, exactly. Perfect. And so, any question about that? So this is very true. The antibody secreted by a particular plasma cell shares the same antigen specificity than the BCR on the B cell precursor that generated such plasma cell. And why does it make sense? Why it makes sense that then the plasma cell secretes the same antibody, that the same soluble form of the BCR in the precursor cell? Yes?
Does it have to do with the MHC? Presenting the antigen to it?
No, not that way. Let's think differently. But thank you for trying. But like maybe you want to think like what are the purpose of secreting antibodies? Yes. Exactly, right? Because the antibodies need to fight the pathogen that is being invading your body, right? And we went through all this process of clonal selection of B cells that are specific for the pathogen, with the ultimate goal of generating antibodies that will bind the pathogen, because only if they bind the pathogen, they will be able to neutralize the pathogen, they will be able to opsonize, they will be able to fix the complements in the context and destroy the pathogen we are interested in destroying, right? I mean, if we have an infection with measles, we will select B cells that recognize, let's say, the glycoprotein of measles, right? And then we will clonally expand these B cells, right, that have this BCR. And then after they are clonally expanding, they become the effector. The effector equivalent of a B cell is a plasma cell that is antibody factory. And so it will produce this very same antibody that was in the BCR, in the B cell precursor. And that makes sense because this is the very same antibody, the very same antigen binding site that in the first place recognized measles and selected this B cell for clonally expanding. There's no use, and it's not helpful at all to have a B cell that recognizes measles, right? Clonally expand this B cell, right? And then generate a plasma cell that produces any antibody, you know? And what I'm going to do with these antibodies, I need to fight measles. Make sense? Okay, that's how you need to think about things. It needs to make sense, okay? Promise me that you will try. And then ask help if not. And help me out to, you know, pose questions that help other students to think, to make sense about things. I would love, and you know, I'm going through college with my son now, so I encourage him to do the same. Make study groups, talk about, meet at a coffee shop and talk 2 or 3 or 4 and talk about the slides and how it makes sense and ask questions to each other. That's how you will remember the most and get the most, and you may get friendships in the meantime. Okay, so E, CD4 T cells enhance the microbicidal activity of macrophages and enable differentiation of B cells into plasma cells. Correct. So the CD4 T cells are helper cells. They will release cytokines and express molecules that enhance microbicidal activity in macrophages and also will enable the differentiation of B cells into plasma cells. And we have a lecture that is T cell B cell cooperation, so we will not go into the details of this. But I want to ask you a question. So again, so it makes sense to you. So how the CD4 T cells know which macrophage and which B cell they need to help? They will be helping any macrophage, any B cell. How do they know who they should help? Yes. MHC class 2. So that's how, remember the trait? That's how they see, okay? The CD4 T cells needs to see the peptides derived from the pathogen or whatever antigen they are specific to in MHC class 2. Otherwise, they don't see, okay? And so now, will they be in a context of an infection? Will CD4 T cells, so again, we will be dendritic cells, parallel to what we just said about the B cells, right? Dendritic cells will be presenting the pathogen peptides and selecting and priming T cells that are specific for MH, in this case CD4s, that are specific for MHC class 2 with a peptide, correct? Okay, so now you go through all the trouble to clonally expand these CD4 T cells specific for the peptides, let's say, again, the example with measles, right? Specific for MHC measles peptides complexes, okay? MHC2 presented misal complexes. That's what the T-cell TCR will bind for these CD4 T cells you clonally expanded. Okay? Same page? Okay, so now this is a helper, and it's already a factor, right? It can be a Th1 and be capable of helping macrophages, or can be TFH and be capable of helping B cells. So now, which B cell and which macrophages will go to the tissue to help? Or will help? The ones that are infected with measles or the ones that are not infected with measles? Okay, macrophages that are infected is a tricky question, but macrophages that, the ones, anyone that present an MHC with measles peptide for which the TCR is specific. So those are the B cells and macrophages that will be helped. And you were absolutely right. It's through the MHC2 presenting the peptide, so the ligand to which the TCR is specific. Makes sense? And that makes sense because now, imagine in the case of the macrophages may have uptaken virus or be infected with virus, but has the virus inside in the vesicles, and we will see how these viral peptides are now presented in MHC class 2, also MHC class 1, but for this case, relevant MHC class 2. And so the macrophage will have MHC2 with a misal peptide, and the T cell that we expanding is specific exactly for that complex. And so that's how it works. And it makes sense because, again, as we were just saying with the B cells, it doesn't make sense to expand. You know, we have expanded a CD4 T cell that is specific for misal peptides presented in MHC2. We have gone through all this process for clonally expanding, and you know, it's useless. It's now this effector T cell will help anyone, right? It will help the ones that are presenting MHC2 with a misal peptide. And that is exactly what we want, because we want it to help the macrophages that have the pathogen. And same thing with the B cells. It will help the B cells that have the peptide with MHC. And the B cells that will have the misal peptide with MHC are the B cells that have a VCR that bound the virus and uptaken the virus. And so now they have the virus peptides inside and can present viral peptides in MHC2. And that's how the CD4 T cells know which B cells they need to help. They need to help the B cells that have the VCR that is specific for measles, so have uptaken the measles virus. Make sense? Any question? Yes.
Just for clarification. So
yes.
Let's say like a macrophage takes up a microbe. It can have multiple MHC2 that has different pieces of microbe.
Absolutely, yes. It can present many different peptides. Yes, absolutely. The excellent question, and we will learn more about that. But that you are thinking perfectly right? Like the virus may have many different peptides. Even the viral glycoprotein have many different peptides. Those peptides will go into MHC2, but there will be a variety of peptides in the surface. So there are many CD4 T cells with different specificities that can help the same macrophage. Excellent, good thinking. Okay, any question? Okay, let's move on then. Yes?
Do macrophages absolutely need T cells to fully degrade pathogens?
Oh, that's excellent. Not always. They are phagocytic cells and they can degrade, but when they receive help, they are better. And so for some pathogens and some microbes, it may be enough. For other ones that may have evolved means to, for example, inhibit, there are mycobacteria that inhibit the fusion of the endosome and the lysosome, and that's how they evade the destruction. For these ones, it's essential to have CD4 help. Okay. Any other question? Okay, so let's move on to the next one. And so here, just to be clear, the false, so the answer that you should select here is C, okay? And so we'll move to the next one. The other small advice, if you allow me, that I give to all of you is that you're making a huge effort to come here, right? You're waking up earlier, you're coming here. You could say, I just come to click, whatever, I don't care, I just get the points. But since you are here, try to do it right, number one, and try to learn and take the most from the effort that you're doing, not just getting the points for the clicker. My tiny advice for you. Maybe you already knew. Okay, so I will stop this one now, so make sure you voted. And so it says, which of the following statement is correct? Macrophages can present antigens to T cells. That's correct, right? We were just saying that. T cells can activate macrophages. That's correct. T helper 1 will activate macrophages. Macrophages resides in the tissues, and this is correct. So, and monocytes, remember, are the blood precursors. They will go to the tissue and differentiate into macrophages. Okay, so all of them are correct. So the one that you should have selected is E. But there was a question, yeah.
Yeah, or just, I can't see the 5 flipped slides on the canvas.
Oh, okay. Okay, I'll do that right now. So why don't you, I'm sorry about that. So this is still lecture 4 slides. But while you guys do this one, the next one on your own, I will make sure the lecture 5 flipped classroom shows up in Canvas. Okay, probably it's unpublished. It's there, I'm sure, but maybe it's not activated. Okay, so this question, go ahead, and I'm starting the poll there, okay? Okay, we'll stop it now. Okay, most of you voted D. So, okay. So there should be there now all the slides from, the flipped classroom. Okay, so most of you voted D. And let's see, which of the following explain why we need to get a flu shot each year? Inability to develop a vaccine against the current influenza virus. This is false because we said we have billions of specificity, and our immune system can generate a response against all these billions of specificities, right? Our innate immunity against influenza virus is compromised. That's not true. We can respond to a virus. It has the pumps. This is influenza is a single-stranded RNA, so it will be recognized by toll-like receptor 7. It will also generate double-stranded RNA, so it will be recognized by toll-like receptor 3, RIG-I pathway, etc. So we can mount an innate immunity against influenza, correct? So this is correct. C says our adaptive immunity against influenza is compromised. That's not true. Again, we have billions of specificity and we can respond. D says new influenza variant strain require a new adaptive immune response. And so this is correct. And this is because we learned that how viruses, and especially RNA viruses that have error-prone polymerase, they change, right? And in the case of influenza, we will learn in the vaccine lecture how it can further change by genetic rearrangements. And so it's true that the influenza variants strains change, that circulate change every year, and it's like a new virus in a way, right? It's different from the one that we had immunity. And so basically that's how the virus persists in the population. And so you could consider it a similar but different virus, right? It's not completely changed, but it has changed enough that our previous immunity is not fully efficient. And that's why we need to generate immunity against the new antigens that the virus has now, that the current circulating strain has. And that's how, the way to do that is to vaccinate, to get these new antigens, and now mount an immune response with memory. For vaccine is very important, this long-term memory that is a characteristic of the adaptive immunity, so it protects us against the circulating virus that has these new antigens. Makes sense? Yes, 2 questions. You can go first, she was first, and then you. Yes.
Is it possible for either innate or adaptive immune system to become compromised against one specific virus, or would it just be the immune system or parts of the immune system as a whole?
So I think that when it gets compromised, and these are immunodeficiencies, and we will learn some immunodeficiencies throughout the course, it's a defect in the immune system, and so it will not respond to many pathogens. When it gets compromised, the immune response gets compromised against a particular virus, is because this virus or pathogen has evolved means to evade the immune response. For example, we learned how herpes viruses have means to downregulate MHC class 1 antigen presentation, right? And so that makes it more difficult. And then there are other viruses that inhibit at different steps the type 1 interferon response. Almost all viruses have evolved means to inhibit type 1 interferon response in the cells where they replicate. And so these are, and then it's never absolute, and we also have in many ways countermeasures for that. And so that's the answer to that question. Okay, your question.
I was just wondering, how come there aren't a ton of different strains of influenza that all need their own unique vaccine? How come we only need one vaccine?
Oh, that's good. The one we get, good question. I know a little bit about it. And so what I know is that the strain, there will be 1 or 2 strains that circulate, and we get a mixture of this strain each year in the vaccine, the circulating strains. And I think that this is predicted based on what has been circulating in the South Hemisphere, or the last strains that were circulating in the past year. And that's, I think, close to what is the correct answer to your question. It's not that that there are hundreds of strains circulating at the same time. And I don't know the value. I'm sure there there is something that, an explanation that makes sense to why there is 1 or 2 or 3, that they are circulating and not more. Probably because the viruses also compete for the niches, but I honestly don't know what is the correct answer for that. Okay. But it's a good news because then we can put these strains that are circulating in the vaccine and generate protection against the circulating strains, and so we're protected for that particular season. Yeah, okay. Any other question? Yes?
So when you get an infection after getting the vaccine, you generate B memory cells. So how would that tackle the infection afterwards? Does it differentiate into something or create an immune response?
Oh, that's excellent. So how you get protection from a vaccine, right? And so, yeah, you get the vaccine, and you should be getting the flu shot. And so you get the vaccine, and then you get an infection by influenza. And so how it protects? Well, there are different ways. It depends on the vaccines. But always we'll be using one of the mechanisms that we kind of mentioned already. So, and it will be always mediated by an adaptive immune response that may empower innate immunity, but the one that protects is the adaptive immunity through different effector mechanisms that we learn. So some and most vaccines, I would say, that are existing and are effective are the ones that protect through antibodies. Opsonization, neutralization, most of them neutralization, at least one component is neutralization, but also opsonization and complement fixation, and also, through natural killer cells, but antibody mediated. And I think that that's a challenge because somehow we haven't generated, and that's the challenge because not all pathogens can be just protected with antibody. And so, because HIV, for example, mutates very quickly, and so, if we have an antibody in circulation for HIV, it will not protect because the virus will mutate quickly and it will disregard that antibody, right? And so, there is a lot of effort trying to generate better vaccines that generate protection through CD8 T cells. And it's a challenge, but but it's an excellent question. Yeah, thank you. Any other question? Okay, so let's do the next one. And so I will start the poll. Oh, wait, we did. So the correct answer here is D, right? Like in this previous one. And now we're doing this one. We are doing this one. Wait. Sorry, we are doing this one, okay? Make sure you are voting because something happened with the base. So make sure you're voting, okay? So vote again. Can you vote? No? It's working for the phone, yes, but the base, I see that.
I don't know if it has anything to do with it, but the slide that we see in the phone is the previous question.
Okay. That doesn't matter because it counts your vote. I'm more worried that some students are. Yeah. Okay. I will have to answer, we will have to start the vote again for this one. Okay, sorry about that. So now should be working for everyone. So make sure you vote. Okay, make sure you vote and I'm gonna stop it, okay? Okay, everyone voted. Okay, I'll stop it now. Okay, says, which of the following is not a way in which the adaptive immune cells empower the innate immune system? Dendritic cells traveling to draining lymph nodes to present antigens to T cells and B cells. So this is not a way in which the adaptive immune cells empower the innate immune system, correct? This is the innate immune cells that are going there then to prime the T cells, okay? So this is the one we should select, and yes, A is the correct answer. So B says antibodies activating the classical complement pathway. We already talked about that. Activation of macrophages by CD4 T helper cells. We already talked about that. Opsonization of pathogens by antibodies leading to enhanced phagocytosis. We already talked about that. Okay? All good? Okay, perfect. So, any question? We'll do the next one then. Okay, so let's stop it now. So make sure you voted. Okay, I'll stop it. Okay, so which of the following molecules function as opsonins, which means enhancing the uptake by phagocytic cells? And so C3b, we learned that can do that, the complement factor C3b. That's true. C-reactive protein can do it as well. And remember, the C-reactive protein also is the one that will activate the lectin pathway. So the mannose lectin and the C-reactive protein activate this complement through the lectin pathway. But they can also act as opsonin, okay? And then last is antibodies that can also act as opsonin, of course, because, and they can act as opsonins, the antibody, using the FIB or the FC fragment. The antibodies, right, they have two parts so this is the heavy change, and this is the light change, correct? So heavy change, let's put it here. This is the longer one is heavier, the heavy change, and this is the light change, correct? but at the same time, okay, so let's see if I have a color. No, but at the same time, there is a part of the antibody that it will be, because these are identical, right? And so 2 identical light chains and 2 identical heavy chains. And there is a part of the antibody that is variable region, the variable region, right? Yeah, somewhere here. And so this part will be the antigen binding site, and this part would be the antigen, the variable light change and the variable heavy change, but this is the variable part. And this is the antigen binding site, and this is the antigen binding site. And these two are exactly the same, right? Are identical, the antigen binding sites on each side, correct? Because they are identical light chains and heavy chains, so they will make, when they couple, they will make exactly the same sequence of amino acids. And so, okay, so in addition to that, that's the most important thing you should know. So in addition to that, based on some treatment with enzymes, the antibodies were dissociated in different parts, in 2 different parts, and I don't care much about the experiment, but you know, it's a nomenclature that you need to know. And this part cuts the antibody here, and so this is here, right? And so basically, this gives rise to a part on the antibody that is called FC, and this is the one that you need to know. And this part, after the enzyme, will be the FIB, and basically you will have the FC, that is this part. This is only after this enzyme was cutting the antibody, and the FIB, which would be like this part. And this one, remember, is the one that has the antibody binding site, the FIB. But this one, the FC, is very important because it has the biological activity. The antibody never existed like that. This is an experiment. Okay? The antibody is not cut like that in nature. Okay, this is just an experiment, but that is what gives rise to the names, which is what is important is the name. So that this part of the antibody refers as FC, okay? And this part of the antibody is the one that will bind to receptors. So many cells have, like macrophages, for example, they will have FC receptors. Okay, and so now they will bind the antibody through the FC. Okay, will bind to the FC portion of the antibody. Okay, and this FC portion is what gives the antibody the biological activity. And the FIB portion is what gives the antibody the specificity, which antigens they will bind. Okay? So, but this biological activity means is good at opsonizing, is good at fixing complement, is good at crossing epithelium, etc. Because that will determine which cells they will team up with to exert their function. Okay. Yes. And this is part of the other lecture. We'll talk a little bit, but yeah. Yeah, please, go ahead.
The B cells are the ones with the FC receptors?
Many cells have FC receptors. B cells, they do have FC receptors, yes. And we will learn more about the different FC receptors. Yes.
I might be getting ahead of the lectures here, but why would a B cell have an FC
Beautiful. Okay. Yes, hold on. I'm going to say this. Some Fc receptors are inhibitory, but you know that's it, and we will learn about that. Yeah. And we can talk more in office hours if you wish. You had another question? Or, yeah, you do? On... Yes, please.
Just based on what we talked about, the peptide enzyme, which was brought up during an experiment, gave way to the names that determine the region's names, and that FAB gives the antigen specificity.
The FAB is the region that contains, is this part, the upper part if you want, and it's the region that contains the antigen binding site, so will determine the antigen specificity. You are absolutely right.
And then the FC is the area for biological activity that binds to the receptors of whichever cell that is doing whatever.
Yeah, excellent. Okay, cool. Okay, let's do the next one, okay? Yes.
I actually have another question. So for FC and FAB, is that only like nomenclature that we give to antibodies, or is it also apply to antigens?
No, antibodies only. Okay, so can we stop this one? Make sure you voted. Okay, I'll stop it now. And says the region of the heavy and light change of the B cell receptor. Remember, the antibody and the B cell receptor are the same, except that the B cell receptor has the transmembrane region. The region of the heavy and light change of the B cell receptor forms the pathogen-specific binding site, okay? Which region from there will be, we just said, is the variable region, right? Could say the FAB contains the antigen binding site, and that would be correct as well. But here is the variable region. The is the correct answer, and most of you answer right. The variable region of heavy and light change of the B cell receptor forms the pathogen-specific binding site, where the constant region anchors the molecules on the cell surface, right? And this is in this lecture, and these variable regions that has the antigen binding sites is very variable. It has also hypervariable areas, right? That are 3 hypervariable areas that are the CDRs, that are the ones that will be in actual contact with the antigen. And then again, this area of the antibody is constant, right? The rest of the antibody is constant, so there is not much variability among different antibodies that we have in our body. And that makes sense because we want variability in the antigen binding site, so we can have billions of specificities, correct? Okay, next one? Yes, let's do it. Okay. Oh, so this was the last one? Okay, and do watch the movies, okay? So what we'll do is we'll do the next one, the first one, in the next flipped classroom that I had here already open. Okay, so we'll do this one. So the first one in flipped classroom slides for lecture number 5. Okay, I'm gonna stop this one, okay? Okay, so it says, what is false about antibody structure and generation of B-cell receptor antibody diversity? There may be things that we haven't learned here. Let me see. Antibodies are globular proteins, that's correct, that they specifically bind to foreign molecules, also known as immunoglobulins. So that's true. I would say that antibodies can also bind to self molecules, and that's how we have autoimmunity. You know, like in lupus, we have tons of antibodies against our self DNA. But the way in which it's written, it doesn't exclude that possibility. So it's true that antibodies bind to foreign molecules as well, and so I would say this is true, okay? B says antibodies are made by B cells that have differentiated to become plasma cells. And that's correct. We have just talked about it, okay? So that's correct as well. C says at a given time, sorry, this is a typo. At a given time, each B cells makes one and only one type of antibody. And then this is correct. And so most of you answered it correctly. Any question about that one? Okay. This will become even further clearer when we learn how antibodies are encoding and gene rearrangement in a couple of lectures. D says each antibody is a copy of its BCR, including the transmembrane region. Okay, so that's the false, right? Because it doesn't include the transmembrane region. Otherwise, it wouldn't be a soluble antibody. So that's very clear. And some of you said all are true. But I think it's clear, right? Probably it was a little bit of a confusion. And that reminds me, read carefully the questions, okay? So your exam reflects your knowledge, okay? So this is false because it says including the transmembrane region, okay? So I'm telling you because there are several students that answer this one as correct. Yes?
Sorry, I know you mentioned that solubility is like tied to antibodies, but did you happen to mention exactly why they need to be soluble?
No, I haven't. But yeah, it's a good question. It's a little bit of why it would make sense to me, at least. So if they are soluble, they can now reach the tissues and they can use these Fc receptors to empower innate immunity. Or they can fix complements but not kill the B cells that are attached to the antibody, and then the complement would lyse the B cells, for example. That is how it makes sense to me that they need to be released. Yeah. No, thank you for your question. Yes? Yes. No, the antibodies. The antibodies. The antigens are made of whatever. We don't even know. You know, there may be antigens that we haven't come up. They are, especially for B cells, the B cells, because they bind native antigen form, they can bind to, very different structures, okay? There's, again, and that's why it makes sense we have these billions of specificity because we need to respond to everything, okay? T cells are more restricted because remember the T cells recognize antigens, they only see them when they are presented in the MHC molecules that will hold the peptides, will be this tray with the peptide, okay? So peptide derives from proteins, so T cells only recognize antigens derived from proteins. Okay, any question? Okay, so I will, is it helpful for you, the summary? Should we go through the summary? Okay, so we'll go quickly through this summary because there are things we have talked about it now or in previous lectures. And then I want to make sure that we finalize the clicker questions, or at least that we make more progress with the clickers, okay? So where is our immune system? And we talked about already about primary lymphoid organs, bone marrow where B cells developed, and thymus is where T cells develop, and secondary lymphoid organs like lymph nodes, spleen, gut-associated lymphoid tissues where are the payer patches as well, that are the meeting place, right? Is where the dendritic cells will bring and the lymph will bring the antigens, and where naive T cells and naive B cells will be constantly recirculating, okay, to favor this encounter. And we put this example with the UCSD students, remember? And only 2 students will recognize the foreign presence, and so how we can favor the encounter. And so the secondary lymphoid organs have these B cell zones, B cell follicles, and we will learn more how when B cells are activated, there is another region that is called germinal center region in these B cell follicles, and we will learn about the B cell, the germinal centers a lot. And then they also have the T cell zone, that is where T cells are. And when T cells need to cooperate with B cells, they will go to the TB border. And we will learn that. We have a lecture just to study this cooperation. An antigen can arrive via different path to this lymphoid. It can arrive through dendritic cells. It can arrive through the lymph. But the spleen, for example, is not connected to the lymphatics. And so it will arrive through the blood. And in the payer patches, there is will arrive across the epithelium through these M cells. Okay, so in different secondary lymphoid tissues, they all have similar structure, a B cell zone and a T cell zone, but the way in which the antigen gets to the lymphoid tissue is maybe different. Yes.
I just wanted to confirm B cells and T cells can also encounter the pathogen while they're circulating, right? It's just that the meeting places makes it easier for them to.
That's correct. Yeah, that's correct. But it's very unlikely that that will happen, because also when they are circulation, there is faster speed and yeah. Okay, any question on that? Yes.
Sorry, can you just specify a little bit more on GALT and its context?
Yes, GALT is gut-associated lymphoid tissues, and that includes some isolated lymphocytes in the mucosal of the gut, but also involves some more organized structures that are called payer patches. And these payer patches have B cell zone, T cell zone, but also they are underneath the epithelium. And so, if this is the epithelium, the payer patches may be here, and again, they will have the B cell zone and the T cell zone, but then this is the epithelium with all the epithelial cells here. And then they will have these cells that are specialized, that are called M cells, and these ones are uptaken antigen from the lumen of the gut, right? From the lumen of the gut, and will bring these antigens, allow these antigens to get in contact to the T cells. And in all lymphoid tissues, we also have the dendritic cells. Remember, the dendritic cells are in tissues, in non-lymphoid tissues, and they can migrate through the lymph, but they are also in the lymphoid tissues, okay? So the dendritic cells will uptake these antigens and will present them to the T cells. Make sense? Yeah? Okay, so then we have general roles of antibodies and its structure, and this is what we just talked, so I'm not going to repeat this. So it's the variable light change, the variable heavy change, remember? The variable light change, the variable area of the heavy change, okay? Then we have another part that is the constant light change. The light change also has a part that is constant, right? Like this part would be the constant part of the light change. And the constant part of the heavy chains is bigger, right? It's all this part. Okay? Yeah. Okay, there are nice cartoons, and you may want to rewatch or check the slides for the lecture, or rewatch the video. And then we already talked about FC and FIB fragments. Okay, what are the hypervariable loops or CDRs, complement determinant regions? And we already talked about it's the parts of the antibody that are in contact with the antigen. How and where does antibody recognize antigens? And we talk about is in the epitope, right? It's the part of the antigens that are bound by the antibody or the TCR. Okay? Any question about that? Yes?
I just want to check, a CDR is like something else with hypervariable.
Yes. Affinity versus avidity, we went into this in the pre-recorded video. This concept of affinity is the strength of one antibody binding site, okay? And the avidity is the strength of many antibody binding sites together. And this is relevant for IgM, for example, because we learned IgM has low affinity, but IgM is pentameric. It means that there is a polypeptide that is called J chain that brings together 5 molecules of IgM. Okay, so it's like 5 antibodies acting together. And that benefits, has 2 advantages. One is that now the avidity, together this pentameric form of IgM, increase avidity, right? And so it can bind with more strength to the pathogen. Okay, so that's one advantage. And what is the other advantage? Yes? Absolutely, and that makes IgM very, very efficient at fixing complement. However, there is one disadvantage. What is it? Too big. Too big, yes. And what is the problem with being too big? The extravasation, right? So it's not good at extravasating. And so it stays mostly within the blood. Any question about that? Okay, so we also learned in this lecture that there are different flavors or isotypes for the antibodies that are determined by their constant region, okay? By the constant region, this part, the constant region is what will determine the biological activity, but also the isotypes, right? And basically it's the sequence of amino acids in this region, right? Which amino acids are here, and particularly in the FC regions, because that will determine if it is able to team up with a macrophage or team up with a mast cell, or what are the activity, right? Or will fix complement, etc. So the 5 isotypes are IgM, IgD, IgG, IgA, and IgE. IgD is not secreted soluble, it's always in the membrane of the B cells, and really is not clear what is its role. So we will not be paying much attention to IgD, mostly because so far there's no known biological role for it, and so we don't understand it much, at least. IgM, we just talked. IgG is good coming subtypes of IgG, but overall IgG is good at opsonization, neutralization, and is good at extravasating, and so it's very abundant in our blood, but also in tissues. And the reason why it's very abundant is because, and we'll learn more about this, as the cells, in general, is produced after IgM, right? IgM is the first one to be produced. And because it's produced after IgM, has been refined and their specificity, and we will learn about this. And so the IgG will start, the antibodies as they fight the pathogen, and we will learn about this, they are refined a little bit, their specificity. They evolve throughout the recognition of the pathogen in a process that we will be learning. And so IgGs produce more in secondary responses and later during a primary response as well. Remember, the primary response is the first time we encounter an antigen. Towards the late phase, IgG will come up. And then in the secondary response as well. IgG is also good at crossing placenta. And then we have IgA, and it's the typical mucosa antibody, especially when it's dimeric, that is basically bringing through a shape change two IgA antibodies. And it's really good at crossing epithelium, and so it helps brings antigens from the lumen into the mucosa, okay, by crossing the epithelium. And then we have IgE that we will learn more when we learn the allergies and towards the end of the lecture, but the IgE act by bringing pathogen and activating mast cells and that produce histamines and other factors that help fighting helminths, but also are involved in allergic reactions, okay? And we'll learn about that as well. Okay, any question? Okay, so then I would like to go to and do more clicker questions. So let's do this one. Okay, I'm going to stop it now. So most of you answer C and says, which of the following statements about an antibody is false? Each antibody has two identical heavy chains and two identical light chains. Correct, we just said that. In contrast to BCR, an antibody does not have transmembrane region. True, we just said that. Each light chain is composed of two variable regions, and that's not true. And most of you, this is the correct, though, so this is the false one, C. And then remember, each light chain, again, this is the light chain here, has only one variable region and one constant region, okay? Is that clear? Okay, so the other one, this says each heavy chain is composed of a variable region and a constant region, and that's correct. Okay, so the correct answer is C. Okay, any question? Okay, we'll go to the next one then. We already did this one, right? Okay, we'll do the next one then. Okay. Okay, I'm going to stop it now, and we can discuss. So it says IgE is the most abundant antibody in the body. No, remember, IgE actually is unique by having very, very small amounts, okay? But also have the highest affinity, sorry, I'm confusing myself, the highest affinity in general. IgM is the only antibody that can be found before isotype switching. And that's true, because in the naive B cells, and we will learn this, naive B cells express IgD and IgM. And IgD is not secreted, but IgM is secreted. And so that's why it's the first one to be released, because it was there even before the antigen. The B cells will produce the BCR in the form of the IgM isotype, okay? And so then there is a process that is called isotype switching, which is when the B cells get help from the T cells, and that is very important. So isotype switching requires T cell help, and remember, this B cell now has the BCR with the IgM, but now can receive help from a T cell through the MHC molecule that is presenting the peptides in the tray, right? And will receive help from the CD4 T cells. And that will initiate isotype switching, that we will learn how it happens molecularly, but basically the IgM, the constant region, will be switched, not the variable region. Only the constant region will be switched of the heavy change, only the constant region of the heavy change. So only this part, okay, will be switched in the isotype switching. And that will now generate, depending on how is the switch, and that will determine by the factors that the CD4 T cells will release, will change into IgG, from IgM to IgG, or from IgM to IgA, or to IgE, okay? And that will change the biological activity of the antibody that is produced, right? The antigen binding site will be the same, because the isotype switching only changed the constant region of the heavy change. Is that clear? Is that clear? Two things about isotype switching that you need to remember. It changed only the constant region of the heavy change, okay? And it requires CD4 T cell help. Okay. By changing only the constant region of the heavy change, it doesn't change the antigen specificity, because it will not even touch the antigen binding site that is in the variable region, correct? And also, by changing the constant region of the heavy change, it will change biological activity, because biological activity is determined by this FC, the region that is in the constant region of the heavy change, okay? Yes, two questions I think that I have there.
So, isotype switching must happen before the antibody can be secreted?
No, like the B cells, you will have B cells in different stages, right? Some B cells will differentiate quickly into plasma cells and will start producing the antibody that they had already, and that's IgM, right? And that's why IgM is the first one to be produced. Other B cells will still be B cells, not plasma cells, and will have the BCR and the MHC2 and receive help, and they switch, and now they generate plasma cells that produce IgG, for example, or IgE or whatever. Yeah. And another question, I think.
I just had like one clarifying question.
Yes.
You mentioned biological activity. Can you give some examples of like biological activity, like what kind of changes we can see?
Yes. Biological activity is how the antibody functions, right? How the antibody is gonna destroy the pathogen, for example. And so, for example, one biological activity that we already learned is opsonization. And so, for example, IgG is really good to opsonize, because the macrophages that we were just talking about have Fc receptors that are recognize the Fc of the immunoglobulin G, okay? And so we'll Fc, and they are called Fc gamma receptors. And the reason is because it's the Greek letter for G is gamma. And so the constant region of the heavy change is often referred with Greek letters. So for IgG, the constant region is gamma. Okay, for IgE, the constant region is epsilon. For IgA, the constant region is alpha. And so this will be a receptor for gamma. Okay, Fc gamma receptor means that it recognizes the Fc in the constant region of the IgG. Okay, and then, for example, mast cells, they have Fc epsilon receptors. And so they recognize the constant region of immunoglobulin E. And these ones produce, for example, histamines. And then histamines will induce itching. And so that's how they will eradicate helminths, for example, or will induce itching in allergy, or also induce blood permeability, vascular permeability, and so it will induce edema, and we will learn more. But these are the difference. So if an antibody, an IgM switch to IgG, then whatever is bound to this will trigger uptake by the macrophages. If it switches to IgE, whatever is bound to it, it will trigger histamine, itching, for example. So it's very different, the outcome, depending on what the antibody switched to. Make sense? Okay. Okay. So IgM is the only antibody that can be found before isotype switching. And that's correct, because IgD is also there, but it's not being released. So it's the only antibody, that's correct. So B is true. A is true, and so the correct answer is, let's see C. Although IgM generally has low affinity, and then it has higher avidity due to its pentameric form. Remember, affinity is for the strength to which one antigen binding site binds to the antigen. And avidity is all together, okay, as a molecule. And this is a pentameric molecule. Okay, so the correct answer is D, okay? So, I want to do, let's see if we can do, no, it's already 9:18, so we will not do this one, and also I bribe you to go to the discussion section. So Shob will answer, help, and other IAs will help with this question and this question in the discussion sections starting today. Okay, perfect. Thank you, guys. Marina, how are you?